Morning students, today we'll discuss about biotechnology principle and processes, chapter 11. What is actually biotechnology and what are the processes we have to apply for this bioengineering or chemical engineering? You can see it is the techniques that is using live organism or enzymes from organism to produce products and processes useful to humans. Means biotechnology deals with the techniques where the live organisms or enzyme from the organism to produce the product and processes useful to humans. So here we are using some live organism or we are using some enzymes also for producing some useful products. In case of suppose in that sense we can use the car making, bread making or wine making. That is also microbe mediated process, and that is a old form actually of the biotechnology, but it is in a new day or new form. It is used in a restricted sense. How it is now to achieve the, we can say, the same on a larger scale or genetically modified organism. So this is for just like the test tube baby, in vitro fertilization. These are DNA vaccine, so defective to for correcting defective genes. So these are actually part of the biotechnology nowadays. Now, European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. So in that sense, what is the definition here? The definition is according to EFB, European Federation of Biotechnology, the integration of natural science and organism. Okay, integration of which one? Natural science, organism and organism, cells, parts of thereof, मॉलिकुलर एनालॉग्स और फॉर प्रोडक्ट एंड सर्विसेज. कुछ प्रोडक्ट के लिए जो इंटीग्रेशन हो रहे किसके बीच? नेचुरल साइंस एंड द ऑर्गेनिज्म, सेल्स, पार्ट्स देयर ऑफ एंड मॉलिकुलर एनालॉग्स फॉर प्रोडक्ट्स एंड सर्विसेज. दैट इज कॉल्ड एक्चुअली biotechnology. Now, what are the principles we are using for biotechnology here? First one is the genetical engineering and second one is the chemical engineering. What is the genetical engineering? Two techniques we are using here. Techniques to alter the chemistry of genetical material, just like the DNA or RNA. The techniques which you are using for changing the or alternation of the chemistry of genetical material, that is actually called the genetical engineering. To introduce this into host organism and thus change the phenotype of the host organism, that is called actually genetical engineering. When you are changing or altering the DNA or RNA, nucleic acid or deoxyribonucleic acid or ribonucleic acid there, and their introduction into the host organism and thus change the phenotype of the host organism, that is called genetical engineering. And then one more condition one more technique is coming also in this biotechnology, that is chemical engineering. What is the chemical engineering? A maintenance of sterile, microbial, contamination free ambience in chemical engineering process to enable the growth of only the desired microbes or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Means in genetical engineering, what we are doing? We are altering or changing the DNA or RNA, and we are introducing that altered DNA or RNA where? In the host organism. That is the first step, and that is called genetical engineering. Chemical engineering, not only to introduce the host organism, but also we have to get the production. So how to get the production? So we have to maintain the sterile environment or ambience in chemical engineering process, so that अगर sterile environment को maintain करें, contamination free environment को maintain करें, तो we will get the means to enable which one? A growth of only the desired microbes. जिस host organism में आपने introduce करवाया DNA या RNA को, उसी होस्ट ऑर्गेनिज्म की डेवलपमेंट और ग्रोथ, वेल डेवलप्ड ग्रोथ के लिए हमें क्या करना चाहिए ना? मेंटेनेंस ऑफ स्टेराइल एनवायरनमेंट्स आर नीडेड देयर. Okay, so that the DGR microbe or eukaryotic cell we can get in large quantities for the manufacture of कोई सा भी बायोटेक्नोलॉजी का प्रोडक्ट, that is antibiotics भी हो सकता है, vaccines भी हो सकता है, enzymes भी हो सकता है. Main motto of our biotechnology to produce these products just like the antibiotics, vaccine and enzymes. For that we have to alter the DNA and then we have to introduce into the host organism and we have to let the host organism to multiply or to grow in a successful manner. For that we have to maintain the sterile and ambient environment. So first one is genetical engineering. उसको जेनेटिकली आपको इंजीनियर करना है. और नेक्स्ट वन इज द केमिकल इंजीनियरिंग बाय व्हिच वी हैव टू मेंटेन द स्टेराइल एनवायरनमेंट सो दैट द ग्रोथ ऑफ द माइक्रोब्स or we can say the eukaryotic cell, that host in that sense we can say can grow in very large quantities and we can get the product in huge quantity. That's all. Now we are coming to the conceptual development. What is the conceptual development is there? Now first one, the traditional hybridization procedure used in plant and animal breeding very often lead to the inclusion and multiplication of the undesirable gene. ये हो गया क्या, जो hybridization process is going on, इसमें एक जो drawbacks है. What is the drawback here? Now undesirable gene also along with the desirable gene they are included. Inclusion or multiplication मतलब included होगा then multiply होगा. The process we will discuss after. So that inclusion and multiplication मतलब आप डिजायर जीन को इंक्लूड किए बट डिजायर जीन के साथ अनडिजायरेबल जीन ऑल्सो इंक्लूडेड हो गए इन केस ऑफ ट्रेडिशनल हाइब्रिडाइजेशन. ठीक है. द टेक्निक ऑफ जेनेटिकल इंजीनियरिंग व्हिच इंक्लूड द क्रिएशन ऑफ रिकम्बिनेंट डीएनए, यूज ऑफ जीन क्लोनिंग एंड जीन ट्रांसफर ओवरकम दिस लिमिटेशन. कौन सा लिमिटेशन ना, जो अनडिजायरेबल जीन इंक्लूड हो गया था, दैट लिमिटेशन will be overcome by which one? By this genetical engineering which includes which one? Creation of recombinant DNA, use of gene cloning and gene transfer. ये तीन process यहाँ पे involved है. And then so allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. आपको अगर if we are going for the hybridization procedure, there along with the desired gene undesirable gene also included as well as multiplied. ये दो जो term बार बार आ रहे हैं ना, that will clear more further because we'll discuss these two terms more here. One is inclusion, another is multiplication. But in case of the genetical engineering, we are going through recombinant DNA, we are going through gene cloning, we are going through gene transfer. So in that condition, isolate and introduction of only one set of desirable gene is allowable here. So undesirable genes are not introduced in this target organism in this genetical engineering process. Okay, now we are coming. So a piece of DNA which is somehow transferred into an alien organism, how it is possible to transfer a piece of DNA to another organism. This process of DNA would not be able to multiply itself. This piece of DNA would not be able to multiply itself in the progeny cells of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. मतलब simply जब that piece of DNA simply it is single, at that condition it cannot be multiplied. But when it is integrated or when it is introduced into the genome of the recipient, it may multiply and be inherited along with the host DNA. Okay. So now, so in that sense we can say how it is actually introduced into the host DNA there. And here it is coming, this is because the alien piece of DNA become the part of a chromosome which has the ability to replicate. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for the initiation replication. Therefore, for the multiplication of any alien piece of DNA in an organism, it needs to be a part of chromosome which has a specific sequence known as origin of replication. बहुत ही important line this one. For the multiplication of any alien species of DNA in an organism, it needs to be a part of chromosomes which has the specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called cloning and making multiple identical copies of any template DNA. So origin of replication is also there, as well as cloning process is also going on. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule, how the recombinant DNA molecules are produced here, and the construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. What is plasmid here? Autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium, that is a bacteria, and in that bacteria plasmid also present there. In that plasmid, the gene coding for antibiotic resistance that will be introduced, and it is actually discovered by which one? Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring the antibiotic resistance. The cutting of DNA at specific locations become possible with the discovery of so-called molecular scissor, that is restriction enzyme. The cut piece of DNA was then linked with the plasmid DNA. This plasmid DNA acts as a vector to transfer the piece of DNA attached to it. And you probably know that mosquitoes act as insect vector to transfer the malarial parasite into human body. Now I am coming to the point here, how it is actually transferring. First, suppose we are saying here restriction enzyme. First, suppose I am taking one bacterium here, and in bacterium I have taken that one extra chromosomal genetical material that is called plasmid is present, and one genetical material is present here, and one extra chromosomal genetical material is present here, that is called plasmid. This is actually working as vector to transfer which one? Na desired gene into the host. So it is actually small piece of DNA. It is small piece of DNA. Okay. This one is step first. Okay. Now what we are doing here? This plasmid is actually working as which one here? Vector. So now this will be cut in a specific site by the molecular scissor, and this scissor is known as which one? This scissor is known as restriction endonuclease. What is the name of this scissor here? Restriction endonuclease. Okay, restriction endonuclease, that we will learn also afterwards. This is one enzyme. So we can say here this plasmid cut by this plasmid, cut by which one? Class by particular enzyme. The enzyme name is here restriction endonuclease. So this is actually step 2. This one is step 2. सेकेंड स्टेप में क्या हुआ ना, दिस प्लाज्मिड को हम लोगों ने पर्टिकुलर जो पॉइंट में कट किया. नाउ इन दैट, बिकॉज दिस प्लाज्मिड इज वर्किंग एज अ वेक्टर, and so plasmid cut by an enzyme that is called molecular scissor, and that is called restriction endonuclease or restriction enzyme. Now we are coming for the particular desired gene we are introducing. Suppose one desired gene is here, and that desired gene is suppose insulin, and that we are introducing here. Okay, so that desired gene is introduced here in that section where we have already cut it. So there only desired gene introduced here, and that is मान it is human insulin, है ना, it is human इंसुलिन मतलब हम लोगों ने यही बोला ना कि एंजाइम प्रोडक्शन करना या एंटीबायोटिक प्रोडक्शन करना, तो यहाँ पे एंजाइम प्रोडक्शन होता है, एंजाइम इज प्रोड्यूसिंग है, तो ह्यूमन इंसुलिन जीन है ना, विच वॉज दी व्हाट इज डीज ओरिजिन, क्योंकि हमें इंसुलिन बहुत सारा अमाउंट में प्रोडक्शन करनी है, तो हमें वी हैव टू गो फॉर दिस जेनेटिकल इंजीनियरिंग. फर्स्ट वन इज जेनेटिकल इंजीनियरिंग मीन्स वी आर मॉडिफाइंग द डीएनए. वी कैन सी हियर दैट डीएनए इज मॉडिफाइड हियर, and then we are introducing this DNA into the host cell to get the multiple product. So insulin, human insulin gene is inserted into where, into plasmid, है ना, कहाँ पे इंट्रोड्यूस किया ना, प्लाज्मिड में इंट्रोड्यूस किया. एंड दिस वन इज स्टेप 3 है ना. स्टेप 3 के बाद स्टेप 4 में क्या हो जाएगा ना, इंजीनियर जो ये जो है ना, इंजीनियर प्लाज्मिड नाउ इंट्रोड्यूस्ड, इंजीनियर प्लाज्मिड नाउ इंट्रोड्यूस्ड. कहाँ पे इंट्रोड्यूस्ड हुआ है ना, इनटू न्यू बैक्टीरियम. एक न्यू बैक्टीरिया में आपने क्या किया ना, इसको इंट्रोड्यूस करा दी, इसको एंटर करा दी. सो नाउ वन मोर न्यू बैक्टीरिया इज कमिंग, एंड यहाँ पे ऑलरेडी उसकी जो जेनेटिकल मटेरियल है और बाकी जो अपने इंसुलिन प्रोड्यूसिंग जीन जिसको इंट्रोड्यूस कराया उसको भी हम लोगों ने यहाँ पे एंटर करा दी. ओके, सो दिस इज कॉल्ड होस्ट सेल. सो दिस इज न्यू बैक्टीरियम. ओके, दिस वन इज व्हिच वन, दिस वन इज न्यू bacterium. Next one, what is coming now? Now this bacteria, this is the host one. तो आपने ये desired gene को यहाँ पे introduced कराया और that is actually genetical engineering we have done. This is called recombinant DNA. कहाँ पे who is working as vector here? This is the plasmid. This plasmid is working as vector here, and in a particular site it is cut by restriction enzyme, and there we have introduced which one, a new मतलब desired gene which we want to get in a multiple way. So now this recombinant DNA, or we can say that this genetically engineered जो vector that is actually introduced into a new host, and now this new bacteria क्या है 20 सेकंड में ये मल्टीप्लाई कर सकते, तो 20 सेकंड के अंदर ही आपको बहुत सारा मल्टीप्लिकेशन आपको मिल जाएगा, मतलब बहुत कम टाइम के अंदर आपको ह्यूज अमाउंट में इंसुलिन प्रोडक्शन हो जाएगा. नाउ व्हेन इट इज इंट्रोड्यूसिंग, व्हेन इन बैक्टीरिया डिवाइड, सो वी कैन राइट डाउन हियर द बैक्टीरियम डिवाइड्स and producing which one, insulin. ठीक है, and producing insulin. तो अभी क्या हो गया, ये बैक्टीरिया जितना डिवाइड करेगा बहुत सारा इसकी बैक्टीरिया दे आर प्रोड्यूसिंग, बिकॉज विद इन 20 सेकेंड दे कैन प्रोड्यूस ह्यूज अमाउंट ऑफ बैक्टीरिया. तो साथ ही साथ क्या हो जाएगा ना, इसके साथ ही साथ आपको इंसुलिन क्रिएटिंग और प्रोड्यूसिंग जो जीन है दैट ऑल्सो विल बी प्रोड्यूसिंग अलॉन्ग विद दैट, है ना, that also will be producing along with that. So this is actually called the cloning procedure, है ना. तो क्लोनिंग प्रोसीजर हम लोगों ने लर्न किया. एक तो हमने लर्न किया रिकॉम्बिनेंट डीएनए. देखो सब में क्या है, इंसुलिन क्रिएटेड जीन जो है दैट विल बी प्रोड्यूसिंग हियर, एंड इंसुलिन कैन बी इंट्रोड्यूस्ड और प्रोड्यूस्ड है. सो ह्यूज अमाउंट ऑफ इंसुलिन प्रोड्यूसिंग जीन हमें चाहिए था, तो हम लोगों ने क्या किया ना, जेनेटिकल इंजीनियरिंग के थ्रू और केमिकल इंजीनियरिंग के थ्रू, इसको ही हम क्या बोलते हैं कि दैट इज कॉल्ड बायोटेक्नोलॉजी, बाय व्हिच वी आर एक्चुअली इंट्रोड्यूसिंग the science into the organism and to get numeral or multiple amount of the product here. तो प्रोडक्ट कौन कौन सी प्रोसेस में हो सकते हैं ना, प्रोडक्ट मे बी इन द फॉर्म ऑफ एंजाइम्स, मे बी इन द फॉर्म ऑफ वैक्सीन्स, मे बी इन द फॉर्म ऑफ एंटीबायोटिक. तो यहाँ पे हमने एंजाइम को प्रोडक्शन कराया दैट इज इंसुलिन. तो इसलिए प्लाज्मिड को हम लोग रेस्ट्रिक्शन एंडोन्यूक्लियस के थ्रू हम लोगों ने कट किया स्पेसिफिक साइट में, और उसी साइट में हम लोगों ने किसको इंट्रोड्यूस करा दिया ना, वो हमारे जो डिजायर्ड जीन था, तो दैट इज ह्यूमन इंसुलिन. उसके बाद हम लोगों ने इसी जो रिकॉम्बिनेंट डीएनए प्रोडक्शन हो गया, प्लाज्मिड इज वर्किंग एज अ वेक्टर एंड दिस इज कॉल्ड रिकॉम्बिनेंट जीन. दैट रिकॉम्बिनेंट डीएनए हम लोगों ने इंट्रोड्यूस कराया न्यू होस्ट में, एंड व्हेन दिस होस्ट इज कीप ऑन मल्टीप्लाइंग, सो यू कैन गेट द insulin producing gene in a huge amount. That is the process actually. And now we are coming last part of this. So last part is that there are 3 basic steps in genetically modifying an organism. What is that? Now identification of DNA with desirable genes. जिस जीन को आपको सेपरेट करनी है उसी DNA को आइडेंटिफिकेशन करना है. Introduction of the identified DNA into the host, है ना। होस्ट के अंदर कौन सा मॉडिफाइड डीएनए, जो जेनेटिकली मॉडिफाइड डीएनए हम लोगों ने इंट्रोड्यूस करना है, एंड मेंटेनेंस ऑफ इंट्रोड्यूस डीएनए इन द होस्ट एंड ट्रांसफर ऑफ द डीएनए टू इट्स प्रोजेनी। तो डेफिनेटली ये 3 स्टेप्स है। पहले डिजायरेबल जीन को सेपरेट करो एंड देन इसको इंट्रोडक्शन कराओ आइडेंटिफाइड डीएनए के साथ इनटू द होस्ट में, एंड देन वी हैव टू मेंटेन एंड इंट्रोड्यूस डीएनए इन द होस्ट एंड ट्रांसफर ऑफ द डीएनए टू दिस प्रोजेनी. Next, we will discuss about the tools of recombinant DNA technology.